Today, we'll be going over Punnett squares, pedigrees, and inheritance, which is a part of the GCSE biology specifications. But before we get started, let's get familiar with some key definitions. Number 1, DNA, or deoxyribonucleic acid, which is a chemical that carries all the instructions an organism needs to grow, reproduce, and function. Number 2, a gene, which is a small section of DNA in the chromosomes that codes for a sequence of amino acids to make proteins. A chromosome, which is a very long strand of DNA that is coiled around histones and normally appears in pairs. Number 4, an allele, which is a different version of the same gene that codes for small differences. Number 5, a genotype, which is the collection of alleles that determine the characteristics of an organism. Number 6, a phenotype, the characteristics seen on an organism, which can range from anything between hair color and genetic diseases. Number 7, diploid, which is when a cell has two sets of chromosomes, and this would normally be most of our cells. And haploid cells, when cells only have one set of chromosomes, and these normally only occur in our sex cells, which is our sperm and ovum. Now to put this into context, we started with nucleotides, which are the building blocks of DNA. A section of DNA such as this, which codes for a protein, is called a gene. Then the DNA is coiled around histones, which are a type of protein. Then histones with the DNA attached eventually condense into chromosomes. And these chromosomes are stored in the nuclei of our cells. As you can see from this picture, diploids have 2 times the amount of chromosomes as haploid cells. And 2 haploid cells, which are sex cells, can fuse into a diploid, which then becomes the first cell of an embryo that eventually grows into a baby. Humans have 22 pairs of autosomes, which are the numbered chromosomes from 1 to 22, plus a pair of sex chromosomes to make a total of 23 pairs of chromosomes. Now let's introduce Punnett squares, which are named after Reginald C. Punnett. These are used to figure out the genotypes of children when 2 organisms reproduce. Chromosomes appear in pairs, since we get one set from each of our parents, and so alleles will appear in pairs as well. Using this knowledge, we can try to find out which alleles the children will have from the ones that their parents have, because each parent will give one of their chromosomes to the child. Alleles are said to be dominant or recessive, with the dominant allele always being the one that gets expressed in the phenotype. What I mean by this is that if there is both a dominant allele and a recessive allele, the information on dominant allele will be expressed over the one on the recessive allele. And so this means that a dominant allele only needs one copy to be expressed, while recessive alleles needs 2. When an organism has 2 of the same alleles, it is said to be homozygous, with homo meaning the same. And when an organism has 2 different alleles, it is said to be heterozygous, with hetero meaning different. Here's an example of a Punnett square using human sex chromosomes. Here we have a mother with XX chromosomes and a father with XY chromosomes. First, you want to draw a table kind of like this, with 3 by 3 squares. And then you want to split up the alleles of the parents. So here the XX is split into these 2 squares, and the XY is split into these 2 squares. Doesn't really matter where you split them into, as long as the X and the Y are next to each other, and these 2 are next to each other as well. Now, then we move on to these white squares, and these will be the squares for the children. So with these, you take the allele from the corresponding column and row, and put them together. So this one comes from the X and the X, this one from the X and X, this one from the X and the Y, and so on. And using the data inside of the white squares, which are the squares for children, we can see that it is a half and half split between XX and XY chromosomes, which explains why we have roughly an even split of male and female people in the population. Alright, let's try a practice question now. The allele for white fur in a guinea pig is recessive, while the allele for brown fur is dominant. A homozygous white fur guinea pig mates with a heterozygous brown fur guinea pig. What are the chances their children will have white or brown fur? So first let's label the alleles, with the dominant allele being labelled capital F and the recessive being lowercase f. Always write the dominant allele as capital letter and the recessive as lowercase, but the letter you use doesn't really matter. And then using the information we get from the question, let's try and figure out the alleles of the parents, which we need for a Punnett square. Since one of the parents is a white-furred homozygous guinea pig, it must have the allele for white fur, and 2 of them since it is homozygous. And for the brown fur guinea pig, it must have 1 allele for brown fur, but the second allele must be different from the brown fur, so it must be a white fur. And so the alleles of the parents must be recessive-recessive and recessive-dominant. So we're going to split these up and then write them into a divided Punnett square, like this. And just doing what we did before, let's take the parents' allele from each row and column and combine them to get the alleles of the children, like this. And from this we see that half of the offspring will have dominant recessive alleles and half of them will have recessive alleles. This means that half of them, the dominant recessive, will have brown fur, and the other half, with the recessive will have white fur. And so it has a 50% chance of being either white or brown. Okay, let's do another practice question. Cystic fibrosis, a disease that causes the lungs and stomach to produce mucus that is thicker and stickier than normal, which causes lung infections, constant coughing, shortness of breath, and poor growth, is genetic and caused by a single allele. Individuals with homozygous recessive alleles are affected with it. Bob and Ali decide to have a child. Bob has cystic fibrosis while Ali does not, but in a genetic test, Ali finds out she has heterozygous alleles. Find out the chance of the child having cystic fibrosis. Again, first we want to label the alleles. So we're going to have capital C for the dominant healthy allele and lowercase c for the recessive cystic fibrosis causes allele. From this, we can try and figure out the alleles of the parents using the information we have in the question. Now, since Bob does have cystic fibrosis and is caused by homozygous recessive alleles, we can label him as recessive. Now, Ali has heterozygous alleles, which means she must have 2 different alleles, so she must have dominant and recessive. From this, we can try and draw the Punnett square for the children, which should look like this. And looking at a Punnett square, we can see that half of the children have dominant recessive alleles, and half of them have recessive alleles. This means the half with dominant recessive alleles will not have cystic fibrosis, while the half with recessive alleles will have cystic fibrosis, and therefore there's a 50% chance that a child will have cystic fibrosis. Now, this is about as hard as questions like this will get, so here are a few tips for working out any similar questions. First, the most important thing is to make sure you know the genotypes of the parents, since you need those to draw the Punnett square. Drawing a Punnett square itself isn't too hard. All you have to remember is to take the row and the column of the parents. Number 2, try to use all the information you get in the question to figure out the genotypes. And remember the difference between homo and heterozygous, since they like to test that a lot. And also, sometimes you might need to go back a generation, to, like finding out the alleles of the grandparents to figure out the genotype of the parents. Now let's have a look at codominance. Some genes can be codominant, meaning 2 or more genotypes can be expressed in a phenotype. Examples include pink flowers, which comes from the codominance of white and red alleles, and speckled hens, which comes from the codominance of black and white alleles. However, the example we're going to use today is human blood groups. So the allele for blood group O is recessive, while the allele for A and B are codominant. So when humans have both the A and B allele, the blood group is AB. But just like before, someone with a dominant plus recessive allele, such as A plus O, will have blood group A. And so codominance really only matters when 2 codominant alleles interact. Now let's have a look at an example question. 2 people, both with blood group AB, have a child. What blood groups can a child have, and what is the ratio of the probability of possible groups? First, just like before, we're going to split up the alleles of the parents, so we get A and B at the top and A and B on the left. And then we draw the Punnett square, and from this we can see that 1 out of the 4 will have A and A, and so the blood group will be A. 2 out of the 4, so half, will have A and B, so the blood group will be AB. And 1 out of 4 will have B and B, and so the blood group will be B. This asks for the ratio to convert the fractions into ratios, and the ratio will be 1/4 to 1/2 to 1/4. And finally, to make the ratio a bit simpler, we want to times everything by 4, so we get whole numbers, and eventually we get 1 to 2 to 1 of blood groups A to AB to B. Next, let's have a look at pedigrees. Pedigrees are diagrams that show the phenotypes of multiple generations, so it's kind of like a family tree but with genetic information, and they can show the inheritance of genetic traits. Horizontal lines, like these, show the connection between two people. Vertical lines coming down from horizontal lines, like this, show the children of the two above. Sex of the people is represented by shapes, so males often shown as squares, and females shown as circles. Affected individuals are shown in a different shade, so in this case, they're shaded in grey. And also sometimes a carrier of a gene without the phenotype is shown as well. So if someone has the gene but doesn't show the traits, they are labeled as well. All of this can kind of vary, but they essentially show the same information, and most of the time in exams they will give you a key, so it doesn't really matter. Again, let's go through an example question. Huntington's disease is a neurodegenerative disorder that is caused by the dominant allele H. The pedigree below shows a family with some affected individuals. Shapes shaded in black represent an affected individual, while white signifies an unaffected individual, and squares signify male, and circles means female. So first question is, Sally doesn't have Huntington's disease and is in the second generation. Find her on the pedigree. Now, if we look at the row of the second generation, we can see that there are 3 females, and only one of them is unaffected, so this must be Sally. Question number 2, find the genotypes of Sally, her sister, and her parents. Since we know Sally and her father are unaffected, their alleles must be double recessive. Now we want to find out the alleles of the mother. Now, since the mother does have Huntington's, she must either have dominant or dominant recessive alleles, but we're not really sure which one she has. So to figure this out, we want to draw the Punnett squares of each combination of alleles. And since we know that Sally is unaffected, we want to look for a Punnett square where the children's boxes have double recessive alleles. So this one must be the correct one. And so using this information, we can also try to find out the alleles of Sally's sister. Since we know Sally's sister is affected with Huntington's, we want to look for any alleles in the Punnett square that signify the presence of Huntington's. Therefore, Sally's sister must have dominant recessive alleles. Question number 3, Sally's sister Lily decides to have a third child with Jerry. Find a chance the child has Huntington's. Since we already know Lily's genotype, which is dominant recessive, all we need is Jerry's genotype. He's affected with Huntington's too, so his genotype must be dominant or dominant recessive. Now we can try and find this out in 2 different ways. First, we can look at his parents, one of which is affected with Huntington's and one of which isn't. His mother has double recessive alleles since she is not affected. Therefore, she must have passed one of the recessive alleles to her son, meaning that he must have one recessive allele, so he must have dominant recessive. We can also look at the genotypes of his children. Since one of his children is unaffected, she must have recessive alleles, and one of those recessive alleles must come from him, since alleles are passed down from parent to child. So meaning he must have dominant recessive alleles. Now, using the information we already have, we can try and draw the Punnett square, which should look something like this. And then using the genotypes of the parents, we can find out that the genotypes of the children are potentially 1 in 4 dominant, 1 in 4 recessive, and 2 out of 4 dominant recessive. Since both dominant and dominant recessive will result in Huntington's, there's a 3 out of 4 chance, or 75% chance that a child will have Huntington's disease. If you're not sure how we got to the alleles of Jerry, you can always draw the Punnett squares and check like we did with the previous question. Now let's look at polygenic inheritance. All the examples we went through so far come from one single gene, since it's much easier. However, many traits are a result of different genes working together, and this is called polygenic inheritance. Poly meaning many, so the translation would be something like many gene inheritance. A good example of this is skin color, which is controlled by the amount of melanin in the skin. The amount of melanin, however, is controlled by many different genes with alleles that work together to promote melanin production, which produces a large range of phenotypes or different shades of skin color. Other examples include eye color, height, and weight. You won't really be tested on anything specific on polygenic inheritance, you just have to know it exists and some examples of it. And that's more or less it for this topic. Now for a quick summary, Punnett squares are diagrams used to find the possible genes of a child from the parents. You need to find a parent's genotype, split up the alleles, and find the combinations using Punnett squares. Dominant alleles are expressed over recessive alleles, and in other words, the dominant allele is always seen in the phenotype. Codominance is when there are multiple dominant alleles, and combining them means both traits are in the phenotype. Pedigrees, these are diagrams that show the phenotypes of many generations, and they're structured kind of like a family tree. You want to work through these logically and slowly, labeling any genotypes you know because of the phenotype. Punnett squares are also very helpful with these. And also, polygenic inheritance is when a trait has many different genes contributing to it. Thanks a lot for watching, and I really hope I was able to help. If you want any extra reading or any extra research, some good websites I'd recommend are Physics and Maths Tutor and BBC Bitesize, both of which have very amazing resources. And while you're here, why not check out other vids from CLS in collaboration with Intuit University.